we work on mahi, and the red drum, and they're representatives of the open ocean and the coastal environment. And we believe that both of these species will serve as models for everything else that's going on in the open ocean and the coastal environment. And what we do is we look at everything from the molecular, cellular level all the way up through population levels. So we cover cell function, organ function, intact animal physiology and behavior, and then all the way up to reproduction, interactions between prey and predators, and how that influences populations. And one of the things that we're beginning to do now, we're combining what we know from the lab, but we can measure their metabolic rate very accurately, to the observations we have of what they do in the wild. And we can then, by combining these two things, estimate what their energy requirements are in the wild. So I've now recently calculated that the mahi population in the Gulf of Mexico, the Caribbean, and the Western Atlantic, that's considered one population. There's about 25,000 metric tons of mahi in that area. To sustain their very active lifestyle, they eat about 900 tons of prey per day. So that's one of the things that's coming out of the work we're doing. We're doing this work primarily to understand the oil spill, but every time we do an experiment where we look at impacts of oil, the reference, of course, is an animal that's not been exposed to oil. And the information we get from that experiment is new. No one knows anything about mahi and how they behave, what their physiology is. And we can now start building on that to learn something about the basic biology of these animals, how they interact in their environment, that we would have never known if it wasn't for the deepwater horizon oil spill. So I think some of the highlights are that what we've seen is that in addition to the heart, which is impacted by oil exposure, it's been known for centuries, well, no, decades. We've shown that olfaction, for example, the ability to detect odors is impacted. We've shown that fish exposed to oil during embryonic development have reduced visual acuity. They can't see as well. Those are two things we've shown. We've shown that their social behaviors, so how they interact with conspecifics, is impacted, basically because their central nervous system or their brain is impacted by the oil exposure. We've shown that fish exposed as larvae will make poor decisions when faced with predators, for example. They will make, they'll assume a more risky lifestyle, which of course in the wild is going to mean you're going to get picked off by a predator. So we've learned a lot of things about effects of oil that are perhaps subtle. You wouldn't necessarily see them when you expose, say, a young fish to oil. You can see that, if you expose them to high enough concentrations, they're going to suffer and die. At slightly lower concentrations, you may see the heart suffering from different problems. They don't swim as well, for example. But if you start really looking carefully, you will see that they don't smell as well, they don't see as well, and they make these poor decisions because their brain is impacted. So that's sort of at the organismal and suborganismal level, some of the most exciting stuff we found. I think the example of the bioenergetics of mahi that I just discussed, so the amount of energy required by the mahi population in the Gulf of Mexico is one example of something we had no idea about. And it's something it's not something you just run out and measure. You can't. Like you got to actually start with the individual animals in the lab, do all these careful measurements on an individual, and then scale that up to the ecosystem. And it's important because these top predators, mahi is a top predator, they have a top-down control of the ecosystem. So whatever they eat is going to impact and cascade down the food chain of the ecosystem. But as you also know, mahi is actually a really important prey source for billfishes, for example. Okay. Something that we're really, really excited about and very interested in. And as you know, billfishes, again, are, from a recreational perspective, very valuable. And also from an ecological perspective. And they are actually threatened in many, many areas of the world. So to understand what their prey basis is doing and what's required to sustain their prey basis will position us better to understand, conservation of billfishes. So I think that's one example of something that we did not expect to find, and something that definitely have much broader impacts than just understanding the oil spill. So we're looking at not just the impacts of oil, but also the impacts of oil in combination with other stressors. So temperature, for example, is a very big factor. What we know is that at higher temperatures, the impacts of oil are much more severe. So in a future spill, if it happens at a time of year in an environment where there's a high temperature, for example, we're gonna know that the impacts on the fish, for example, are gonna be much, much more pronounced. And when it comes to, and I know this is a dicey topic, but when it comes to the impacts of dispersants and the decision of whether to use dispersants or not, I think some of those considerations are really, really important. At high temperatures, you want to be a little careful with getting the pHs into solution because they're going to hit the fish really, really hard. So I think that's one thing where we're helped. Another thing we're seeing with the tagging is that mahi actually cover vast different distances when they migrate. And so if you have a spill in the northern Gulf of Mexico, for example, going out and looking at what's happening to the mahi population in just that area may not capture the whole picture because these fish are moving and others are coming in. So just a basic understanding about the biology, not just of the animal, but also the interactions with other stressors, I think position us better to make decisions for future spills.